This lecture we are going to cover enzyme immobilization. We already discussed enzyme kinetics, and then also we covered enzyme inhibition kinetics. While we discussed or while we're learning these two topics, all we assumed is like you have an enzyme and then it combines with a substrate, it forms some enzyme substrate complex, and then eventually you get some products plus your free enzymes back into the solution. So this is the basic assumption of mechanism and kinetics, and then we also saw different ways or different types of inhibitor that can affect this whole enzymatic process. So in all of these assumptions are in mechanism and kinetics, and also the other expression that allows you to to predict what type of inhibitor that you are dealing with. We all assume like enzymes, they are just dissolved in the water, and you also have some substrate dissolved in the water. They come together, they form some complex, which are which eventually get converted into products and leaving your free enzyme back into the solution. But we never talked about like a how you add your enzymes. It's like a freestanding enzymes, or whether these enzymes are sticked to some other material, or whether once you form your product you have your enzyme on the right hand side, whether your product contains product, if the product is in solution, whether it contains enzymes or just pure product. Yeah, so we never discussed about purity, so we are going to cover something related to this topic in this lecture. And before that I want to mention like there are many many enzymatic reactions, like where we do something to convert some substrate into products plus enzymes, but in many processes we will never recover the enzyme from the final product. So your final product will contain some of the enzymes which you used to convert your substrate into your product. So few example are is like one is manufacturing of cheese. They use lactase and lipase. So lactase, what it can do, it can convert your fats into fatty acids. Likewise, your lipase, it can convert your sorry, no, it's not true. Lipase converts your fats into fatty acids, and lactose converts your lactase converts your lactose into easily digestible sugars. And if you go to supermarket, you can see like washing detergents that also contain some enzymes in the final product. In fact, that enzymes helps to remove the stain from your fabric. Yeah? So that enzyme can quickly act on the stains and remove it from your fabric. So these are few examples where we really don't care about where your enzymes are. Where it's normally you leave the enzymes in the final product. But many industrial products that we are going to produce using some enzymes, we expect a high level of purity. Yeah. So once you produced your product, we don't want to see this enzyme in the final product, which means your final product should be free of enzyme. So if that's the case, then you have to recover the enzyme. You have to recover the enzyme from the product. So in this lecture, I know we are covering enzyme immobilization. So I'm just giving a few details like why do we need to do why do we need to even immobilize an enzyme, or what's the motivation behind that. So that's what I'm going to explain in a bit by bit. Yeah. So for the moment, just imagine if you want a pure product, then obviously what you think is like you have to remove your enzyme from the final product. And there are many situations. This sometimes the enzymes which you are using to convert your substrate into product, they may be ridiculously expensive to produce, and also it's and it's also it will be uneconomical to imagine, like you start a batch, you convert your substrate to product, you and then if the enzymes are really expensive, it's not a good idea to leave your enzyme in the product. It's better to recover those enzymes, and then so that you can use use it in the next batch to convert more of your substrate into new products. Yeah, so it also for the purpose of cost or the money involved, we have to recover the enzyme from the final product. And apart from that, during enzymatic reaction, another problem that we commonly encounter is like enzymes tend to agglomerate. So as I said to you, enzymes are molecules. It has this beautiful packet, and your substrate will go into that particular active spot, making this enzyme substrate complex, and you need this to convert your substrate into products plus enzymes. So imagine if you have a lot of enzymes, if they come together, what it happens is like they agglomerate, and then they lose those active packets. So these active packets will be masked merely due to agglomeration. So this is some unwanted event that can happen inside a fermenter. Yeah? So we don't want this one during an enzymatic reaction. So no agglomeration. So so we have to avoid this type of issues. Yeah. So and then and sometimes like 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 enzymes can be really expensive. Yeah, as I said to you. So as I said, if the enzymes are expensive, then you have to recover, and if you have to recover, then you need to imagine, like you have some reaction, you have some enzyme plus your substrate, you get some products plus enzyme, then you have to go through few processes to recover your enzymes. So we call them as downstream unit operations or downstream techniques. So once you produce a product, whatever the operations that you are going to perform, like like a simple filtration or drying or centrifugation, everything we call them as downstream techniques. We are going to cover this in the next lecture in detail. So I'm going to say what are the different downstream unit operations that we are going to perform once you produce a product using some biological reaction. It can be using a live cell or using an enzyme. Yeah. So so if the enzymes are expensive, we should recover, and then we should go for some of these downstream techniques. Few to mention, like filtration and centrifugation. Filtration we can perform using like a membrane filtration, for example, or like reverse osmosis. And and whenever you are going to perform some unit operation like filtration or centrifugation, it involves you need to install those units, and it involves some operational cost plus material handling plus labor cost. So many issues involved, which means it's going to ultimately increase your final product. And another reason, say just you imagine if you manage to form your product, and if you want to separate your product like enzyme plus product, and you want to separate your enzyme from this product, imagine if you just go for a filtration process. As I said, enzymes are large molecules, but still they can pass through your filter paper. Yeah? So which means you cannot practically filter. If you pass your solution that contains products plus enzyme, you will get both product and enzyme in the filtrate. So nothing will be retained on the filter paper, and that's because your molecules are so small it can pass through the pores of the filter paper. So it makes this type like filtration technique is not a viable technique. Yeah. So due to the size of the enzyme molecules involved, but So these are some of the issues when you are going to how do you call, like if you're going to use like a free enzyme, like you take a reactor and you put some enzyme molecules. So these are the issues that you are going to encounter or if you want you can call like practical difficulties or practical problems that we might encounter when we are going to use an enzyme in a dissolved state. Yeah, so what we are thinking is like, you have these enzyme molecules and they are just dissolved in the solution, or if you want you can call it as a fermentation broth, yeah, that contains your product and you are going to convert them, sorry, that contains your substrate and you are going to convert them into products. So so whatever the issues I said are the practical problems that I said, you can avoid them easily by doing one thing. Yeah, it's called enzyme immobilization. So enzyme immobilization can help you to to skip most of these commonly encountered problems while manufacturing some product using some enzymes. Yeah. So I'm just going to quickly explain the advantage. Use like taking this filtration technique as some example. I said to you like if you have a solution with enzyme, you pass through the filter paper, everything pass through. So which means nothing will be retained on the filter paper. You will get your enzyme plus product. But just assume if you load, take a big solid particle, and if you load your enzyme on that solid particle. So what we are doing is like so this in the solution, your enzyme particle freely move here and there, yeah, especially if you are agitating your reactor. But what we are doing now is like we are immobilizing them. So we are going to artificially stick your enzymes onto some larger solid particle. Yeah, for the moment we can call them as a carrier particle because it's carrying your enzymes. And if imagine if your solution contains enzymes attached to this big solid particle plus your products, we can easily filter them. Yeah, imagine this is your filter paper, you can easily filter them because these solid particles are bigger than the pores of your filter paper. So which means you get your pure product in the bottom. So what it makes, like once you immobilize your enzymes on some solid particle, it allows you to perform one of the unit operation very easily, like a simple filtration. Apart from that, it also allows you to obtain your product in purest form. So this way you will get a pure product using one of the simplest technique, filtration technique. Yeah. So this is one key advantage if we decided to immobilize the enzymes. Let me check my notes. Yeah. And normally, for example, filtration is like is less energy intensive. Yeah, it's in fact is one of the cheapest technique. And if imagine if you are not immobilizing your enzymes are tiny molecules, and normally they are okay, they are large molecules, but still they are small enough that you cannot see with your naked eye. Yeah? So they are almost like molecular size, and you cannot use a filter paper. Then you have to go for energy intensive process like centrifugation. Centrifugation is relatively expensive when you compare it with the filtration technique. Yeah, and then after centrifugation you also need to spend some time in removing your supernatant solution from the bottom. Yeah, I hope you all know how to perform a centrifugation experiment. So this is really an energy intensive process. So one way is like by immobilizing enzyme, we can not only get a pure product, but we can also use some simple techniques to to separate enzymes from the product. So this is one advantage of immobilizing an enzyme. Yeah. and And about these carrier particles, as I said, this is industrially used technique. What they use is like they take some solid particles with really very high surface area. Normally they use like a silica material, silica based materials. So each gram of those materials will roughly contain 800 meters squared per gram of your silica. So what they do is like, which means you can load really enormous quantities of enzyme on this 800 meters squared. So in 1 gram of silica, you can load a lots of enzyme molecules. Theoretically, yeah, but practically it may be difficult. But theoretically speaking, you can something like, if you have 800 meters squares of area, you can place a lot of enzyme molecules. So that's the idea.